Designer Babies, Frankenstein Reality, or a Step Towards Disease Prevention? Did you ever wish that you could change something about yourself? Almost everyone has at some point in their life, but has faced the reality that their characteristics cannot be determined by themselves or others. However, this might not remain true for very long. The age of genetic selection, modification, and enhancement is upon us. Scientists are developing technologies that are highly advanced in gene editing, such as CRISPR technology. Through in vitro fertilization, or IVF, where an egg is fertilized in a lab, scientists have been able to perform pre-implantation genetic diagnosis, or PGD, for a few years now, which can determine the characteristics of different embryos. Currently, PGD mainly identifies diseases for the purpose of choosing a disease-free embryo of a couple that is at risk of passing a serious hereditary disease onto their child. Additionally, PGD can also identify gender and a few physical traits. However, soon the list of traits PGD can test for could include physical strength, intelligence, musicality, and more. Since all of these methods are or would be done at an embryonic stage, parents would be the ones deciding which characteristics their children would have. As we are all heavily influenced by societal constructs and media portrayals, which inform us of the most valued and advantageous traits, you and your parents would likely pick similar traits. The benefits of selecting your offspring's traits do not come without consequences, however. Embryo selection outside of disease prevention risks crossing ethical and moral lines, but more serious than that, it risks decreasing genetic diversity and increasing the economic divide. The application of genetic engineering to humans poses an even greater threat to our future, as errors would affect future generations and success would cause extreme discrimination and would directly interfere with evolution, which is what has helped us become the intelligent and complex species we are today. To clarify, I am against genetic engineering of the human genome, but I am also against not allowing for the selection of embryos on any grounds. The only condition under which a couple should be able to have a designer baby is to prevent the passing down of a serious hereditary disease to their child through embryo selection, because genetic modification technology is unsafe and the biological and societal risks of selecting for or altering non-life-threatening traits greatly outweighs the benefits. If allowed to genetically modify or select their child as an embryo, parents will hold the fate of their lineage and future generations of humans in the palms of their hands. Even now, the valued characteristics that are plastered all over the media and affirmed throughout society are leading people towards using colored contacts, bleaching their skin, and abusing performance-enhancing drugs. These practices will carry over to gene modification, and people will begin to choose socially valued characteristics. Lighter skin, non-brown eyes, good-looking, intelligent, athletic, musical, extroverted. Based on the current media portrayal of what characteristics are favored, these are the characteristics people will begin to design their children to have. While I do not believe in the dominance of these traits, these are common trends and pressures that remain present and highly influential in our society, despite the constant battle against them. If these influences reach our DNA, we risk inhibiting the very mechanisms that have brought the human species to where we are today: natural selection and evolution. The driving force of evolution is natural selection, and in turn, natural selection depends upon genetic variation. Genetic diversity is influenced by the random production of gametes, which are the egg and sperm, random mating, the individuals who decide to mate, random fertilizations, the one sperm that fertilizes the one egg, and mutations, or mistakes in the DNA. Embryo selection reduces the randomness of fertilization, since embryos are chosen, not randomly implanted. In IVF, several eggs are fertilized in a dish, and the embryos implanted are chosen out of a group of embryos, deeming the concept of random fertilization predictable and controlled. Reducing the randomness of fertilization through embryo selection does not have a big effect on genetic diversity, because mutations and random gamete production still provide genetic diversity, which are unaffected by embryo selection. In the world of designer babies, the influences of random gamete production and mutations on genetic variation would be muffled by genetic modification. This is because any mutation will be corrected, and the genes in the egg and sperm cells that combine to make the child will be altered, so whatever genes pass down from the parents to the child are considered irrelevant. Through gene modification and societal pressures, there will be a drive to mate with people who are, quote, enhanced, influencing mate selection. The problem is that enhanced individuals will have similar characteristics and therefore similar DNA. If this occurs, our race as a whole will be unable to adapt to our ever-changing environment. This is because natural selection drives evolution by selecting for traits that allow the organism to survive in their current environment, making them the fittest for survival. The fittest individuals will survive, mate, and pass these advantageous traits on to their children, and individuals who die will not mate and therefore not pass on the traits that hindered their chances of survival. The next generation is then better able to survive in that environment. The species as a whole has adapted to its environment. The wider the range of traits present in a population's gene pool, the more likely that one of them will be the trait that allows the species to survive in a new environment. Gene modification would remove mutations and reduce the diversity of traits in our population, making us less able to adapt to changes in our environment. This is especially important considering the current state of our planet, where the ozone layer is being depleted, greenhouse gases are becoming thicker and more dense, global warming is progressing, sea levels are rising, and carbon emissions are increasing. In their article on the morality of designer babies, Mario Sousa and Alberto Barros hold the viewpoint that we will, quote, allow the selection of advantageous traits because the ultimate goal will be an improved species. Furthermore, it may be necessary to scale up the evolution of the human race and secure our survival, which they believe will have no effect on populational diversity. I do not agree with this viewpoint of society in terms of evolution because while our goal is an improved species, evolution also shares this goal. In his book, The Impact of the Gene, From Mendel's Peas to Designer Babies, Colin Tudge states, "The idea that we can improve on our own good cells by fiddling with our genes like washing machine repairmen seems to me not simply hubristic, but ludicrous. It has taken us 5 million years to evolve to our present state. Natural selection really does work and has been a hard taskmaster. It is obviously dangerous to override it. We cannot afford to squander the thousands of subtleties that our evolution has built into ourselves, and in particular into our psyches." Tudge owes our success as a species to evolution and finds the idea of messing with it absurd, and so do I. Adding on to Tudge's remarks, evolution is responsible for making us the most sophisticated and complex species on Earth. Evolution has ensured our survival throughout human history and is much more reliable than experimental methods of gene modification. Also, if we only select for advantageous traits, those characteristics may only be advantageous in our current environment. What happens when the environment drastically changes and a trait that was previously deemed not advantageous and therefore weaned out of the gene pool is now advantageous and critical for our survival? While the effects of designer babies on evolution could be far in our future, we need to consider all of the major effects of embryo selection and genetic engineering, from future effects like decreased genetic diversity to those in the near future, like economic unfairness. Embryo selection is occurring now, while genetic engineering is still in the trial and error stage. However, embryo selection is accompanied with the IVF procedure, or in vitro fertilization. IVF is expensive and will be more expensive than the added cost of pre-implantation genetic diagnosis, or PGD, which identifies hereditary diseases and other traits of the embryo to allow for embryo selection. Gene modification procedures would be even more costly as it is more experimental. This will widen the gap between people of high and low socioeconomic statuses, as the extremely high cost of these procedures restrict the technology to this financial stable class of the society. Sousa and Barros argue that since an economic divide is, quote, already present already the present rule in our societies and everyone accepts this as a normal thing, the widening of the economic divide that would be caused by genetically engineering humans would not change much. While I concede that there is an economic divide already present in our societies, I bring up the point that this economic divide would no longer be based on materials, but on our DNA, our genetics. Today, the factors that cause this gap are money, opportunities, jobs, and living conditions. Unenhanced, people of different socioeconomic classes have the same capacity to excel, while lack of resources and support makes succeeding more difficult for people of lower socioeconomic status. People know that if all those factors are controlled, they are no different than anyone else. With gene modification, not only will people of lower socioeconomic status have less resources to help them succeed, but they will be at a genetic disadvantage. People of higher socioeconomic status will not just seem as though they are better because they have excess resources, they will actually be and be perceived as superior to the unenhanced individuals. This gap will lead to the discrimination of genetically unenhanced children. One day, not being genetically enhanced could be a determining factor on a job or college application. Elitist views could arise in families with multiple generations of enhanced individuals, in which the higher the percentage of enhanced individuals one has in their family, the higher class or caste they would be. This social construct is seen in the Saudi Arabian royal family, and it also appears in the Harry Potter series, where some believed that the more wizards that were in your family, the more, quote, pure blood you were, increasing your worthiness as a wizard. Despite being a fictional series, the books draw parallels to real issues and effectively portray the issue at hand. Even now, the economic divide, discrimination, and the disadvantages of being of low socioeconomic status are serious issues in our society. Their increased prominence due to designer babies would drive our society into chaos and towards becoming a society similar to the one Hitler envisioned in the Holocaust. While gene modification technologies that threaten equality exist, they are not safe, and without completely understanding genetics or the human genome, we risk the safety of human embryos in human applications. Even though the Human Genome Project was completed in 2003, we do not know the exact function of each gene, especially for genes that influence multiple traits. Alternatively, we have also not been able to identify the different genes that influence polygenic traits, or traits that are influenced by multiple genes, for instance, intelligence and personality. Genes usually work in tandem, so until we fully understand all of the genes in the human genome and their functions, there is always the chance that disrupting one gene may disrupt larger signaling pathways, leading to cell damage. In an experiment done on the genetic enhancement of learning and memory in mice, researchers made genetically altered mice, or Doogie mice, by adding an extra copy of the NR2B gene so that they made more of a glutamate receptor, which is known to play a role in the development of the brain. The Doogie mice had higher than average performances on mouse intelligence tests, but an unexpected effect was that they were also more sensitive to chronic pain. This result suggests that in mice, and possibly other vertebrates like humans, memory formation and pain sensation might share components of a common physiological pathway. This experiment demonstrates how the alteration of one gene, even when done correctly, can cause unintended effects on DNA, since one gene can influence several different characteristics. Until our understanding of genetics and our own genome greatly improves, we should not consider using genetic modification technologies on human embryos. Lack of understanding is only one risk factor in using genetic modification technology on humans. The other risk factor is the technology itself. Recently discovered, the most promising gene editing tool is CRISPR technology. Scientists found an interesting set of genes in bacteria called CRISPR, or clustered regularly interspaced short palindromic repeats, which they later discovered codes for an enzyme called Cas9 endonuclease. Cas9 is part of the bacterial defense system and is target specific. meaning that as long as it is used correctly, the tool could be used to find a defective gene and replace it with a corrected version. While CRISPR technology is the most precise tool to date and has had success in experiments using animals, it is not completely safe or error-free, especially when it comes to humans, which is why the NIH will not fund any use of gene editing technologies in human embryos. CRISPR technology was used in experiments to try to protect human stem cells from being infected by HIV, but the results were conflicting. One report was that the cell's repair mechanisms made mistakes when repairing the DNA targeted by Cas9. These mutations happened to prevent the HIV virus from surviving in the host cell, but mutations are unpredictable. How can scientists know that the repair mechanisms in the cell will make the same mistakes again? If they do not make the same mistakes, then the cell may not be protected from HIV anymore, and there may be more negative effects on the cell. The Chinese edited the DNA of embryos using CRISPR-Cas9 for the first time in 2015, but despite succeeding in editing the targeted defect, unintended defects were also produced elsewhere in the embryo's DNA. In most experiments, it seems that the problem is the unintended effects in other parts of the DNA, not the target sequence itself, demonstrating the unreliability and danger of CRISPR-Cas9. Harmful mutations have been a consistent problem with the use of CRISPR-Cas9 until just recently, when an international team of scientists found a way to edit human embryo DNA using CRISPR-Cas9 without causing harmful mutations. In response to these findings, critics are proclaiming their fears of getting closer to achieving designer babies, but the scientists insist that their work is aimed at preventing terrible diseases, not creating genetically enhanced people. Altacharo goes even further, claiming that the constant drumbeat about the fear of designer babies is unwarranted. But I beg to differ. While we are not there yet, I think the fear of the designer baby is quite warranted. Designer babies could come from the misuse of this technology, or by what we might believe to be the proper use of CRISPR. Before experimenting with human DNA, scientists were experimenting on animal DNA to learn more about the workings of genetics, but there's a catch. Almost every animal experiment is done for the purpose of future human application. The Doogie mice experiment involved genetically modifying mice to be more intelligent, which could be applied to preventing learning disabilities, which may be partially caused by mutations in genes controlling intelligence. This could be classified as a genetic disease, and scientists would start experimenting, wanting to learn more about the genetics behind intelligence, which would crack open the door to the future of designer babies. We are well on our way towards genetic enhancement and the reality of designer babies. The groundbreaking experiment, which involved correcting the mutation causing cardiomyopathy by using a new approach, corrected the mutation in more than 70% of the 58 embryos. This would be a good outcome if the risks only applied to the individual embryo. The scientists do not elaborate on what happened to the other 30% of the embryos. The risks may be worth it for an individual, but when editing DNA, one risks the safety of the individual and future generations as well. Any mistake made while editing the DNA of one individual would be passed on to future generations. A mutation could cause the genetically modified individual to develop a new disease that would be passed down to their progeny and their progeny's progeny until the entire family is permanently burdened by this new disease. The risks are too high and could affect too many people. Disease prevention cannot only be achieved through genetic modification. IVF and PGD allow for embryo selection, a method that is far safer than gene editing and does not pose a threat to future generations. Today, IVF and PGD together are used to select the disease-free embryos of a couple that is at risk of passing a serious hereditary disease onto their child. This prevents a genetic disease from being passed from the couple to their child and future generations. Some still argue against embryo selection in any situation because it is unnatural to choose who your baby is. I understand that people may have certain religious beliefs regarding IVF and PGD, but embryo selection gives a couple carrying a serious genetic disease the chance to have a healthy baby and not pass the disease on to their child. It is a fairly safe procedure, as shown by the many healthy children who have been born through IVF and PGD. Not only would embryo selection help to remove a deadly disease from a family, such as Huntington's disease, but it would also relieve future emotional and financial stress on the family. In society as a whole, the overall cost of healthcare would decrease if these serious hereditary diseases were prevented before birth. Embryo selection achieves the same goal as gene modification, but with lower risk to the embryo and no negative effects on future generations. Although there is a fear of creating designer babies through embryo selection, as there is with gene modification, it is much less likely, since the traits chosen are predetermined and are from the parents, not genetically altered. If proper restrictions are put in place regarding the information tested for by PGD, then the traits that embryos can be selected for would be restricted to diseases and traits directly linked to the disease in question. Scientific discoveries in the field of genetic engineering and pre-implantation genetic diagnosis of human embryos are occurring rapidly, and critics are pushing for the government to take action and place certain restrictions on both. Gene modification experiments should not be conducted on human embryos due to the enormously high risks, but should continue to be conducted on animals to increase genetic understanding. This is where you can help prevent the idea of designer babies from becoming a reality. In the end, human application of the methods that would result in genetically enhanced babies depends on legislation. The government must make policy regarding these advances in science, and the people have a say in this. While the current topic of the debate within the overarching theme of designer babies is whether we should perform gene editing experiments on human embryos, policy questions regarding designer babies will progress as technologies improve. If people fight for the purpose of PGD to be disease prevention only and fight against the application of gene editing technology to humans, we can save our society and future from the negative effects of designer babies.